Hi everyone, this is Fahad Mirza and I welcome you to the channel. I have a firm belief that one of the areas AI is going to have huge positive impact is healthcare, especially where humans have reached their limits and only stuff like AI can help out. This is why I am so excited to share with you this newly released model Metagene-1. This model would enable us to predict pandemics. So in this video, we are going to install it locally and then we will see how it works. And before that, let me give you a bit of a technical overview of this model. Now, as I said, this model is a 7 billion parameter autoregressive transformer model, which is called as metagenomic foundation model that has been trained on a new corpus of diverse metagenomic DNA and RNA sequences comprising over 1.5 trillion base pairs. This is specifically optimized to detect anomalies in short metagenomic reads, so 100 to 300 base pairs, making it really well suited for tasks like pathogen detection and biosurveillance. The model's architecture choices, such as its 512 token context length, limit its applicability to complex sequence design tasks, so that reduces misuse risks because that is another huge area where work is being going on. Now, look at this diagram which is on your screen. This is about is how exactly they have sourced their data, which is quite interesting. This data set is sourced from a large collection of human wastewater samples processed and sequenced using deep metagenomic sequencing methods. Unlike genomic models that focus on individual genomes or curated sets of specific species, the objective of this Metagene-1 is to capture the full distribution of genomic information present across the human microbiome. After pre-training, this model has been designed to aid in tasks in the areas of biosurveillance, pandemic monitoring, and pathogen detection. Now, let me introduce you to the sponsors of this video who are AgentBot. AgentBot lets you effortlessly deploy a personalized knowledge bot across platforms like Discord, Slack, and others. It is ideal for open source tech communities and startups that provide user support, and I will drop the link to their website in video's description. Now let's try to get it installed and then we will see how it works. As you can see, I'm using this Ubuntu system and I have one GPU card, NVIDIA RTX 6000 with 48 GB of VRAM, courtesy Massed Compute for sponsoring the VM and GPU for this video. And if you're looking to rent a GPU on very affordable prices, I will drop the link to their website in video's description with a 50% discount coupon, so you are going to enjoy it. Let me start by creating a virtual environment with Conda and then we will install all the prerequisites. And those prerequisites are transformers, torch, and accelerate. Shouldn't take too long to get installed. And now let me launch my Jupyter notebook. And now let's import the model and its tokenizer. And the model is being downloaded. As you can see, there are 6 shards of it, which is a bit unusual because normally these 7 billion parameters models have 4 shards. Anyway, so let's wait for it to get downloaded. The model is downloaded and loaded onto our GPU. Let me show you how you can do the inference or pandemic monitoring, or basically the sequence monitoring with it. So first up, as you can see that we are passing it this input sequence, which is just some sequence here. Now this input sequence is the initial DNA sequence that we have fed into this Metagene model. And then model is using tokenizer to encode it, generating the output as per its own intelligence, and then we are decoding back the result, and then we are printing out the response. So let me quickly run it to see what it produces. And there you go, look at the response of the model, looks really cool. Now this response as per model's card is something which is called as, and it's a hard word, it is called as hexameta inflata 5.8s ribosomal RNA gene sequence, which I have no idea what it means. If you are from this field, please enlighten us and let us know what it means. Um, Google says that it is a species of parasitic protozoan that infects fish and other aquatic animals, and it's a type of microorganism. And then it uses this RNA, which is a type of RNA that makes up large part of ribosomes. But I'm not going to go into that detail. You can search it out and see what is happening here. But the point is quite interesting. If you look at this model, what exactly it is doing, it can really contribute in a very valuable way to pandemic detection and monitoring in several ways. For example, by analyzing metagenomic data from wastewater samples, Metagene can help identify new or unknown pathogens, including viruses, bacteria, and other microorganisms. And this early detection can provide critical insights for public health officials to respond promptly, unlike COVID pandemic. And this model can monitor changes in pathogen populations over time. That allows researchers to track the evolution and spread of diseases, and it can also help predict potential outbreaks and inform strategies for containment. This model can also identify genes associated with antimicrobial resistance, enabling early detection of AMR in pathogens. And this information can guide the development of effective treatment strategies and inform public health policy. It can also be used for real-time surveillance and monitoring of wastewater samples, providing timely insights into the presence and spread of these pathogens. And this rapid detection can enable swift response and intervention, reducing the risk of outbreaks. And I think it can also be integrated with existing surveillance system, enhancing their capabilities and providing a more comprehensive understanding of pandemic risk. So I believe not only this model but similar models can really play a vital role in detecting and monitoring pandemics, ultimately supporting global health security, which is a really lofty and noble task. And really, I can't wait for further research, further models in this area, which I'm more than sure are going to happen. If you go through its model card, not only it gives you a lot of data around the model, but also various benchmarks are there. For example, they also have this gene MTAP benchmark, which you can check, which goes through, which evaluates its ability to produce high quality zero shot genomic representation, which you can check out. So seems really interesting. I will drop the link to it in video's description. Let me know what do you think. As usual, if you like the content, please consider subscribing to the channel. If you are already subscribed, please do me a favor and share it among your network, as that really helps a lot, and that's the only marketing I'm doing. Thank you very much for all the support and have a good day. Thanks.